Thank you for coming. It's my great pleasure to introduce Eddy Rubin as today's Major Issues speaker. Many of you will know of Eddy from his role as director of the Joint Genome Institute just down the road. He actually got his undergraduate degree in physics, and then he got an MD and PhD working on Bucephalus genetics at the University of Rochester Med Center. From there, he moved to the University of UCSF, working genetics from 85 to 88, and then moved to the Lawrence Berkeley labs in 88. He became chair of the genomics division in 97 and director of the JGI in 2001. He's had a big impact not only through his own research, but through stewardship of the JGI. He likes to call himself a consumer and generator of large-scale sequence data. This has really enabled a tremendous amount of comparative genomics. The JGI has had a big impact in terms of not only sequencing several chromosomes in the Human Genome Project, but they went on to sequence all sorts of very interesting organisms, and others that would never normally have been had the traction to get that amount of sequencing done. So that's really enabled a lot of very, very interesting biology. He now has fostered the community sequencing program, so you can apply to the JGI. I don't know if you can talk a little bit about that, but that has enabled a tremendous amount of sequencing of all sorts of organisms, and a number of people at Davis have been beneficiaries of that. So it's a great pleasure today to welcome Eddy Rubin. He's going to tell us about extreme comparative genomics from microbes to Neanderthals.
Okay, great. Thank you very much.
Thank you very much.
It's fun to be here. I've very much enjoyed everyone I've spoken with, seeing old friends and meeting some new ones. Okay, so I'm going to talk about 3 different areas. I'll begin by sort of giving a little infomercial about the Joint Genome Institute. This is a user facility. We're looking for more users, and so I'll tell you a little about what we're doing and how to interact with us. Then I'll talk about 2 areas of science in my lab. I'll talk about our work at developing a set of enhancers covering the human genome. And finally, I'll talk about this fun project, a real high throughput exploitation of sequencing, and that is Neanderthal metagenomics. Okay, so the JGI, this is a large-scale production facility in Walnut Creek. It was built by the Department of Energy to make its contribution to the Human Genome Program. As said, we sequenced our 13% of the human genome in 3 chromosomes. This facility is really it has 110 capillary sequences. It runs 24 hours a day, 7 days a week. It was built for the human genome, for that single genome. I think something that most of you are probably aware of is that we're about to see this enormous shift in sequencing, in that the motivation to developing places like the JGI was that single human genome. Now there's a motivation to sequence individual humans. And there are 6 billion humans on the planet. There's a large consumer market out there. And so this has really driven technology, and technology needs these kind of targets to drive them. This is, if you want to start you want to put out a shingle and say sequencing technology, you can raise very easily venture capital for it. And so right now we have 110 of these, ABI sequencers that are pumping out 70,000 base pairs a run. They're going to be replaced in the next 2 years with machines I'm not sure what they're going to be. But the one thing we're confident about is that we're going to increase our capacity 100-fold to 1,000-fold. And the cost will remain at what we're spending now for that much more sequence. And so therefore there's a different kind of biology that you're going to be able to do with this. And so as I talk about some of the things we're doing, I'll try to mention a little bit where I think things can be going in the future. Okay, so what is the capacity aimed at? And as you heard, we're a user facility, somewhat in the model that, light sources and supercomputers, people put in scientific proposals. The government if the proposals are deemed of merit by the review committees, then we will generate sequence for you. And we cover just about the entire biosphere, except that small niche of mammals. And so we're really driven by bioenergy, bioremediation, carbon cycling. And so if you have projects that somehow in your first paragraph can relate to those topics, those are fair game for the JGI. Don't use the word biomedicine. Okay, and so we've become a fairly complex facility because previously we had a single target, that was the human genome. Now we have more than 300 projects that are running through the facility. Okay, and so we have a lot of users from UC Davis. UC Davis is next to UC Berkeley as a number of users. We're interested in increasing that, and so that's our website. We have various programs that try to find projects that fit in. We can generate sequence for you as well as help you in the analysis. And so we're interested in people that have really interesting science that connects to these mission areas. And frequently we get naive users that aren't used to using genomic data but have very interesting problems. And we provide the infrastructure to help you solve those problems, as well as some people we just provide flat files of sequence for more sophisticated users. And so we have a large portfolio already involved in the process of moving from cellulose to ethanol genomics. We have a whole series of feedstocks that we're sequencing: poplar, switchgrass, maize, parts of switchgrass. Where we have a bunch of organisms that play a role in the breakdown of cellulose, white rot fungus. We're sequencing microbial communities that play a role in the breakdown and conversion of cellulose on the way towards ethanol. Termite hindguts, the microbial communities in the termite hindgut that convert your floorboards into dust. Tamara wallaby force stomach. These are funny organisms that are put forward by scientists, and all the data we generate goes on the web, and hopefully this will become a parts list that people that want to move forward in engineering organisms in the future connected to the mission of bioenergy will be able to exploit. And then we also have organisms involved in the final stop of conversion of ethanol producers. So not only are we sequencing these feedstocks, but we're sequencing organisms that impact on them. And we're sequencing pathogens, because somewhere down the line, if this industry is going to grow, we're going to want to know this information. We're sequencing feedstock symbiotes. Okay, so these are the kind of projects that have come to us from our users. The future. This is the poplar that we sequence. Nisqually 1. This is our reference poplar genome. It was next to the Nisqually River in the state of Washington. The river rose and the tree is gone, but its genome is, in the database. In the future, one is going to want to sequence a forest for looking for natural variation. One is going to want to look for phenotypes in that forest that you want and not spend years trying to map it, but rather sequence it and see if you can see variation. And just as though we're trying to resequence humans, looking for variation that contributes to human phenotypes, the same way we'll be able to, and it's not very far in the future we'll be able to do this. And so this is the JGI. This is the poplar. Though the Nisqually one got washed away, poplars are clonal. And so a branch of that poplar has been planted in front of the JGI, and so we both have its genome as well as a clone of the tree. These are some of the feedstocks that we're sequencing. They're all going to be planted in front of the JGI. We'll hopefully have a botanical garden in not far in the future. Okay, another area of emphasis is microbes and metagenomics connected to these issues. And right now, there are a lot of microbial genomes that have been completed that are on the way to being completed. The JGI has become, the JGI and the Venter Institute and Tiger are really the major sequencers of these microbes. Most of these microbes are onesie, twosie. Someone had an interest in a microbe and put it forth. That's our view of the microbial world. I mean, I think what's going to happen in the future is that one of the projects that we're putting together that's going to be run by Jonathan Eisen from UC Davis is a program we call Genomic Encyclopedia of Bacteria and Archaea. And this is a program to really, to phylogenetically go out there and sequence several thousand microbes and make a reference genome for the entire microbial kingdom. And the background for this is that right now there are probably about 40 phyla of bacteria. Though we've sequenced a fair number of microbes, most of them are in a very limited 3 phyla. But all these others are not. And these are ones that frequently have biomedical interests. And so the hope is in the future is to do deep sequencing along all the phyla, or as many of the culturable phyla. And hopefully there'll be ways to get in some of these unculturable realms. We have many metagenomic projects. These are projects where you go into environments. You don't worry about isolating a particular organism. You sequence everything in the environment, and from that you build the biology from the sequence data. And it's, I think, a way in which sequence can inform you. In much of the world, the planet, we can't culture in our laboratories. This is one window into that part of the planet. Where this is going in the future, right now, we're getting a single view of metagenomes. We're sampling and we're sequencing. And it's like a snapshot. In the future, with these new capacities, one would like to get moving pictures of environments. And so from these sort of stop-motion animations, narratives will emerge that we will not have gotten from a single view. And I think this is where metagenomics will be going in the future, with the ability to sample many times in space or in time a particular environment. Okay, so this is the end of the infomercial. I'm a sequencing guy. Dramatic improvements in DNA sequencing will have a near revolutionary impact on accelerating all aspects of biological research. And you have problems that need sequence, interesting ones, particularly if they relate to energy, bioenergy, carbon sequestration, or bioremediation. That's our website. Come visit us and send in a proposal. Okay, let me move on to the science. One of the challenges of running a large sequencing center, at least during the human genome era, is that each of the chromosomes were divided up, and each center had responsibility for a certain number of them. And so we had responsibility for these 3 chromosomes, and part of that responsibility was writing a paper about them, which was an enormous burden because genomes are not meant to be analyzed, broken into chromosomes. They're meant to be analyzed on a whole genome scale. And so writing these papers, what has become clearly eminent is that we have different capabilities of analyzing sequence data. And there's certain kind of sequence that we're pretty good at reading, and there's other kind of sequence that we just have not a clue to read. And what we're fair at reading is coding regions. We're not bad at predicting where genes are. And I think that if I went around this room and particularly asked the graduate students or postdocs and gave them a genome from anything on the planet, they could do an okay job at predicting where the genes are. And why is that? And there are lots of obvious reasons. We have data sets. We've been accumulating, there's a code, there's a triplet code. We have large EST data sets. We have protein data sets. So we've been accumulating this data, training sets, for a very long time. And from having these training sets, we've developed very good algorithms that are able to sift through long stretches of sequence and identify where the genes are. Okay, the other part of the genome, the non-coding part, we're clueless at being able to extract information from that. Being able to look at, which is 99% of the human genome, being able to go through that genome, identify where the regulatory elements are, we have not a clue. And we really score an F in this regard. And part of the reason, I think, is we don't have big training sets. And so what I'd like to talk about for the next little period of time is development of a large set of characterized gene regulatory sequences. So how do we look through the 99% of the genome that is not coding? And it's a large space, but the availability of a variety of vertebrate genomes have begun to shrink that space because studies have suggested that sequences that are conserved deep in evolution have function. And that relates to coding regions. Genes are conserved. But also this non-coding functional component of the genome also is conserved. And so by doing cross-species sequence comparisons, we're able to shrink this 99% of the genome to a smaller space that we begin to interrogate. And so the approach that we've taken is to look for extremely conserved sequences, one that have been conserved for hundreds of millions of years in evolution, or ones that have been highly conserved. And the 2 categories, one is deep evolutionary conservation, things that have been conserved between humans and fish, that is several 100 million years of evolution. And then things that are called ultra-conserved elements. And these are elements that are 100% conserved between human, mice, and rat for over 200 base pairs. And so these are sequences that are unchanged for the 80 or so million years between humans went their separate ways for rats and mice. Okay, so this is the filter that we're using to sift through the 99% of the genome of non-coding sequence to identify sequences that we think play a role in that may play a role in gene regulation. Okay, the next step is to develop the functional assays. And so the way we, first of all, to get the data, we align the human and mouse genome and the human and fugu genome. And what I'm showing you here is 10 kb of human sequence along the horizontal axis. The vertical axis is the percent identity to mouse there. Here is the fugu genome and the percent identity to fugu. And what you can see is that the exons will frequently stand out as conserved. In fugu, not much else stands out, but these are the kind of elements that we're interested in. In the mouse, we're interested in elements that are 100% conserved. So this is how we filter the space. Then what we do is we place them into a reporter construct and introduce them to a mouse embryo and come back 9, 10 days later, or exactly 11.5 days later, and look for this sequence here driving expression of this reporter construct. And if we see, and we demand that at least 3 of the embryos have it expressed in the identical pattern, and if we see 3 mice with the exact pattern of expression, here this would be forebrain, we will say that this element is an enhancer driving forebrain expression. And so this assay has, this is the antithesis of genomics. It's low throughput, but it has the great advantage in that it makes no assumption about what these sequences are doing, because at this time point we can visualize the whole embryo. And so, we've tested tissue culture systems. This looks at an organismal level where this element is driving expression. And so we've developed a crank. And also over the last 2 years, we've searched for through more than 400 of these elements, testing them for enhancer activity. And what we're pleased is that close to half of them do have enhancer activity. And so the filter is a very good filter, that we're not having to make tons of transgenic mice that don't have activity. And so half the elements we test will generate the same pattern of expression in several different embryos. What's the relevance of the positives? We're not looking for genes. We don't really care what genes are around these elements. We're really just going through the genome and identifying the elements and testing their activity and using evolutionary conservation to identify what elements we're looking at. And one of the things that we see, these elements frequently will drive patterns of expression similar to neighboring genes. And I think that is best illustrated with sonic hedgehog. And so we have one of these elements a megabase away from the sonic hedgehog gene. Sonic hedgehog, remove the gene, you get a variety of abnormalities. We know sonic hedgehog plays a big role in limb development. Okay? People have taken this element and put it in the same reporter assay, and this is a 200 base pair piece of sequence that is non-coding, is conserved between humans and fugu, and you put it in the assay I described. It drives expression in the limb. You remove this element. You lose sonic hedgehog expression in the limb. The mice no longer develop limbs. And finally, in humans, there are humans that have point mutations in the sequence, and they have limb abnormalities, extra digits. Okay, and so this is all saying that this sequence here that conservation identified plays a role in driving sonic hedgehog expression in the limb. Okay, so that's sort of the biological relevance that we associate with these elements. Another thing is that the information that's encoded in these sequences is highly reproducible, the information the functions associated with them. And though here are 3 elements that we would classify as limb enhancers. Okay, and they each give different patterns of expression. One gives this triangular pattern. This one gives this apical ectodermal ridge pattern. One gives this little dot. when we look at 3 different transgenic lines with this element, they all get the same pattern. The same thing here, and the same thing here. And so what we're seeing is that embedded in these several hundred base pairs is this information. People have taken off the web and PCR synthesized these elements, put them in their own systems, and driven expression, the identical patterns that we're seeing here. And so just as we associate activities to certain protein structures, there are activities associated with these non-coding sequences. And now we've begun making large numbers of them, bins that drive similar patterns of expression. Here is, this was our first 160. This is what we have with 400 tested. So now we have a large number of forebrain enhancers, lots of different central nervous system enhancers. We have lots of numbers of heart, eye, limb enhancers. And so there are lots of elements now that drive expression in these specific tissues. And this is a project now supported by the NIH. There's a database. You can go in and get all the experimental data we generate. You can view it in a couple of different ways. You can view it by searching chromosomes or by genes. And you can find elements next to genes that you might be interested in. An example, we're used to looking at the genome of a linear array of genes. And so this is 3 megabase of chromosome 16, and we're seeing the genes that are distributed along that chromosome. What we have now is we've added to that these elements, and these elements drive specific patterns of expression in the mouse embryo. And so if you're interested in a quantitative trait that maps to this interval, and the trait impacts on limbs or one of these, then it would suggest that that element is one that you might think about resequencing in a population. And so it's really a way of shrinking. You'd like to sequence the whole genome, but maybe right now you're going to sequence the gene. You may also, these would provide you with elements that you might want to resequence in large populations with your particular condition. You can also search by expression patterns. And so you click forebrain, and now we have more than 40 forebrain embryos that will come up. You click limb, and now about 25 limb embryos that will come up. You can then zoom in on a particular one, and you can see all the different transgenics that were generated with the same sequence. You can also, each of these embryos sit in a little bottle, and we've had requests for people who have wanted to look more deeply. This is a gross view. People have, this is one that, we had a bunch of embryos that had heart expression, but we've sent the embryos to this investigator who did more deep sections. And what we were calling heart is saying he has a bunch of enhancers that specifically drive outflow tract, ventricles, and endocardial cushions. So you can zoom in. But we have this crank, and so we're looking very grossly at the whole embryo phenotype. But one of the prices of us sending them the embryos is that these histological sessions are now in our database that you can access. So the applications, I think that you now have widgets that will drive tissue-specific expression. The role of regulatory sequences in disease, and I think this is an important area in that anyone that studied Mendelian disorders, you will have people that have a disease, but you just won't see a mutation in that gene that is known to be causative for that disease. And this now gives you regions to begin looking for changes which may be contributing. And globin is a model of this. There are lots of people with globin abnormalities who have mutations in these regulatory elements, and so this is now provides you with a set to look for that. And finally, development of gene regulatory code. This is really a training set. And so here we have now more than 180 enhancers that drive expression in vivo, and you might want to drive expression in a particular tissue. This is now a widget that you can stick next to a gene or a reporter or whatever you want to be driven in that tissue. One of the questions that we were interested in looking at was, can extreme conserved elements be exploited for the rational design of synthetic gene expression patterns? Can we kind of do synthetic biology with the animal? And this is linked to the modular nature of these elements. And this, what we view is that, one of the views of these elements is that if you look at a gene, there are these elements around it, and these elements act in cysts to drive expression of the gene at a particular time and space. And this clearly has been most well characterized with globin. One of the questions we ask, if regulation is truly modular, can it be exploited to build specific expression architecture de novo from existing modules? Can we take a bunch of these modules, put them together, and make a predicted pattern of expression in different places? And so we've begun testing that. And we've taken a couple of the elements that drive very reproducible expression patterns, put them together out of context from separate chromosomes, and seen whether we can get, whether it's additive or whether they interact or whether they interact with each other. And so here are the kind of results. Here we take a forebrain enhancer and a limb enhancer. We take the two elements, we put them next to each other, and here comes the mouse, and we see it's additive. And here's midbrain and forebrain. Put them next to each other. We get a combination. So it's really very, very additive, at least we're seeing in the examples that we've looked at. Put them together, you get both patterns of expression, midbrain, hindbrain, both patterns of expression. Here is two different limb. This is the dot. This is the apical ridge I showed you previously. You put them together, and you get both of them put together. And here's the one, just a very recent result. We want to see how far can we go with this. Can we take 4 of these elements and we get a composite of all of them? And who thinks it won't work? I didn't know. Anyway, so you take the 4 elements, put them next to each other, and you're basically getting the pattern that you saw from any of them individually. And so in summary, there appears to be no signs of interaction interference between these elements. It appears to be highly reproducible. And it supports the extreme modular nature of enhancers, that you can take bits and pieces from all over the place, put them next to each other, and you can get a composite pattern. What can you do with it? You can get predetermined expression patterns for gene therapy. Sometimes we'd like to get something expressed in a specific tissue or a specific time. This allows us to predetermine how we want this expressed and derive it. Also, it's tools for synthetic biology in animals. One could think about, people are doing all kinds of cool things in microbes. One could think about building animals that had certain features driven by predetermined of how and where you want it to be expressed. Okay, so the regulatory development of the gene regulatory code, and this is work another Eisen, whose lab is next to mine in Berkeley, and one of his graduate students, Alan Moses, and Mike is a computational biologist and been interested in looking for patterns in transcription factor binding sites, and so now we've given him bins of sequences that drive expression at specific places. And he basically was interested in going through those sequences and see if he could see features, and then use that to go through the entire genome and see if we could predetermine a non-coding sequence that had specific activity of where it was going to express. And so we started with forebrain, and this was early on when we had a small data set. And so made a training set from all the forebrain enhancers on chromosome 16, to find significant overrepresented words in this training set, then search the entire genome for those overrepresented words, and tested them using this assay. And the test is basically the transgenic assay. All right, so here, this is a small training set. We had 4 robust forebrain enhancers in the set of 84 elements that we had all together. And so 4 out of 84 of these conserved elements drove forebrain expression. We then tested 3,000 Fugu conserved elements that were found throughout the genome, looked for overrepresentation of this forebrain signature, and tested the best 23 predictions. And 5 of the 23 did drive forebrain expression. It's a pretty good result, not a great result. It's encouraging. We've been able to bring about a 3-fold improvement in our ability to predict it. But the really way you'd like to do this experiment is iteratively. See the data, see what works, see what didn't work, then go back. And we're doing that with these studies. But it's encouraging. Important point is that when we did these initial studies, we only had a limited number of forebrain enhancers. And so we were calling this a forebrain enhancer. Here's the histological data from that. And we were also calling this a forebrain enhancer. And here's very different histological data. But now with this big data set, we're now able to pick enhancers that have identical patterns of expression. So we have a much better training set that we hope will be better in making these, in sifting through them and making predictions based upon overrepresented signatures that we see in them. And so here's the site. All this data is available. It's fun to play with. So far, we've looked at 400 elements. We plan over the next 3 years to look at more than 2,500. And also, we invite computational predictions. People go through our training set, make predictions. We're willing to test them. And so, in summary, I think, it's early on in being able to go through the genome and identify, be able to read what is a regulatory element. And so I think we're scoring F. But an important point is that I think this is a midterm grade in that there are new data sets that, the real goal with the human genome is to be able to read it. And not just, and I think there are parts we're pretty good at reading the nouns. We're not very good at identifying the verbs and many other features of the speech. But I think that we're getting ways and tools that will make us much better at this in the near future. Okay, so I'm gonna now turn on to my last topic, Neanderthal metagenomics. And this is the remains of a Neanderthal that was found in southwestern France. It had died about 40,000 years ago. It clearly had been buried. It hadn't just died in the back of the cave, because the soil that was around it is very different from all the other surrounding soil. There were artifacts that were associated with it. We have tremendous fascination with Neanderthals. It comes from our interest. We share a common ancestor. We're very interested in what we came from. All right, so let me tell you a little bit the history of the field and and the history of science is very interesting. So I'll digress a bit and tell you about that. And that's, my interest in them just came from this brute force sequencing, and then as I learned more and more, I got fascinated with how science progresses. And this is an important year. This is the 150th anniversary of the discovery of Neanderthal. And there was, in the Neander Valley in Germany, there was some workers looking for limestone. They blew open the entrance to a cave, and they came upon this pile of bones. And they noticed some features, and the features they noticed was the bones seemed a little more robust than what they were human bones, and they looked aged. They were mineralized. And they brought them to the attention of Johann Fulrad, who was a local naturalist and school teacher, and he got it. And this is the beginning of this field of paleoanthropology. And you have to realize that at this point, there was no realization that humans weren't the hominid species, and that there was no realization that there were species that had went extinct and no longer existed. And so this was really this was information that just didn't fit in the context of what people knew was prior to Darwin. And so Johann Fulrad and a local academic, Hermann Schaffhausen, published a monograph in the Bonn Natural History Society about the Neanderthal, the Neander man. And they got, in the description, they looked at it, they did describe it as a possible ancestor tribe to modern humans. And so they did get one of the concepts of it. There was another monograph published a few years later, a little thicker, had many more printings, and that was Origin of the Species by Darwin. And this spread like wild, no one was really paying attention to the Neanderthal man. People were paying attention to this. This was discussed in coffee shops, but eventually it was the monograph about the Neanderthal was translated into English, and it was read by Thomas Huxley, Darwin's bulldog. And Huxley immediately placed, and this is the plate from his book on the evidence of man's place in nature, and he placed the Neanderthal man right in this step. And he got a little bit wrong with gorillas and chimpanzees, but he overall did pretty good. And this was great. But there was a lot of resistance to evolutionary theory at that point in Europe, not so unlike it there is today. And in fact, one of the great opponents was the father of cell biology, Rudolf Virchow, who and it was a pathologist, and he described the Neanderthal man as a pathological specimen. And he thought it had rickets, he had a fractured arm, and explained the supraorbital ridges that Neanderthals have due to the pain and the frowning of this individual due to his broken arm. And this was his response. And he called the remains an unfortunate deserter from the Cossack cavalry that happened to wander into this cave. And one can't help but think that maybe his view of science, the role of evolution, was somewhat clouding his judgment, because this clearly was found deep, buried under 10 feet of soil, and the Cossacks had been there just 100 years earlier. And so, but science has this natural self-righting process, or that we hope it has this. And what was going on then was that suddenly popping up throughout Europe were remains that looked quite similar to the one that had been found in the Neander Valley. And this is the remain that was a Gibraltar cranium, and this in fact had been found a few years earlier than the Neander Valley remains had been found, but it wasn't described. But it was later on described by George Busk, and it had all the features that the one from the Neander Valley had. And he wrote that even Professor Virchow will not suggest that a rickety Cossack also crept into a cave in the Rock of Gibraltar. And so I think with this, as well as remains coming up all over, suddenly the concept that there was another species, or at least some version of the species that fit these anatomical features of Neanderthals. All right, so what do we know about Neanderthals? And basically we know about them is, a few bones and stones. And that's we know where they were. And they were in Europe from about 300,000 years ago to about 28,000 years ago. What's unique about them amongst all hominid species is that we see no remains in Africa. Homo erectus and Homo sapiens, we see remains in Africa. Neanderthals are only seen in this sort of narrow latitudinal range. And what's believed is that they were some precursor, Homo erectus or Homo heidelbergus, migrated out of Africa, and then this was a period of extreme cold in Europe, and they evolved in response to the climate. Okay, but anyway, that's where we see them. They stretch from the Mediterranean, from the southwestern France, Spain to Uzbekistan. And the other things we know that they seem to live in similar geographical regions of anatomically modern humans about the same time period, and whether they overlap for 5,000 or 10,000 years, in Europe, we're not sure. But basically we know about them is a collection of bones and a few stones. And going through the literature, one can't help but giggle about the theories and hypotheses that are derived from these kind of data sets. And I think there's lots of wonderful stuff, and it's easy to be a genomicist and make fun of these paleoanthropologists, but I can't help myself. There are theories about their religion, about savage brutes are carrying humane, their attire, and the kind of data. And here is the withered arm of the one Neanderthal specimen, and clearly this individual aged after the damage had occurred, and so from this comes the theory that they were compassionate because somehow they cared for this one individual. They see worn down teeth, frontal teeth have been noted in Neanderthal skulls, and from this there comes this theory of a garment industry where Neanderthals processed furs by pulling them through their teeth to attire Paleolithic hunters. And so this is the kind of data that leads to the theories about their behavior. But I think the most definitive finding about Neanderthals is that they're gone. And they probably disappeared about 28 to 30,000 years ago. And one would hope that the glacial will recede and Neanderthal will appear, a frozen one. I don't think so, because it really seemed they were in Europe in a very cold time, and it got warmer. And so I don't think that we're going to see them, as the Alps, lose their snow. And so what we have is maybe a little more than 100, maybe 200-300 skeletons, bits of 200-300 skeletons, and a few associated artifacts. And that's what we have to learn about our ancestor. But I think that we are on the verge of getting another data set. And the data set is their genome. And I think that will provide, it won't be able to tell us about lots of the things that the bones and stones can tell us, but it will provide us another kind of data set to begin to speculate about what they were like. And what particularly is pleasing to me is that I think particularly pleasing to me is that now I've been working very hard to access some of this material, and it's clear that the material is hidden away in museums and in faraway places that no one wants to part with them. And so a very limited number of people get to study the data. Well, I think in the future with having billions of bases on the web, a lot of people are going to be able to look at the data and begin to think about what this ancestor was like. Okay, so the critical questions as one thinks about doing Neanderthal genomics is it possible to recover Neanderthal genomic DNA? And genomic DNA has never recovered from an ancient specimen until just recently. And then one of the other issues is authenticity and contamination. And this is really a crucial issue in that, I love Jurassic Park. And, I love the concept of the dinosaur, the amber and the mosquito biting the dinosaur and getting the blood. And, and there were a couple of papers in Nature and Science that were published on this, but it turned out they were all lab workers' DNA. It wasn't real. And so that's the problem with ancient DNA is that contamination can really fool you. And so dealing with that is one of the other challenges. What can be learned from the Neanderthal sequence? When did modern humans and Neanderthals diverge? That's really a crucial question. Our relationship to them. Is there any evidence of admixture? So see, these are some of the things that we can learn from the sequence. All right, so why is it so hard to get the material? Well, you have a nutrient-rich source in the back of a cave for 40,000 years. Other things are going to try to access that nutrient. And so basically what's there is a lot of microbes and other organisms that have been living off the remains for 40,000 years. And then there's other kind of damage. And so that's why genomic DNA has not been, it's not been studied from Neanderthals until just now. But time is right for Neanderthal genomics. And there are a couple of things that have come together in the last few years that make this feasible. One is that decreasing costs of sequencing, because when you go in there, only a small fraction is going to be what you want. Most are going to be the microbes you don't want. But if you can really sequence deep and fast and cheaply, you can begin to look at billions of base pairs. And if a percentage of that is what you want, then you begin to learn from that. And so that's one thing. Another one is reference genome. You can't, we have bits and pieces of sequence. Now we have a reference genome, so we can place those sequence back on the human genome. And so that's essential for us to begin to think about looking at the Neanderthal genome. We couldn't do that prior to having the reference human genome. And finally, metagenomics. And this is sort of a conceptual strategy in that we previously used to think about genomics. You look at a pure bacterial culture or a piece of flesh from a mouse or a human, and you sequence that. You sequence that human. You sequence that bacteria, or you sequence the mouse. Here you have a mixture, the metagenome, all the different genomes. But metagenomics don't worry about that. Allow the computer to go through it and pull out what you're interested in. So these couple of things come together that enable us to begin thinking about Neanderthal genomics. And so I was thinking about it, and I didn't begin with a Neanderthal. I began with a cave bear. And this is, I developed a collaboration with Svante Pabo, who's somebody who spent his life collecting these ancient samples, and I learned that these Neanderthal samples are quite precious, to promise your firstborn to begin to access one of these samples. And they should be precious because there's a finite amount of them. Cave bears, on the other hand, not so precious. There are lots of them. They died in caves. In fact, last year you could buy a whole cave bear skeleton on eBay. Okay? And the other thing about cave bears, we employ no bears at the JGI. And so we have no problem with contamination. Okay? And so if it looks like bear, it's not the worker, it's the bear. Okay? And so I won't go through the study, but we did a study and we generated a bunch of genomic sequence from a cave bear. And for a variety of ways, we were able to prove it really was cave bear, and it was able to place cave bears in between polar bears and black bears. And, and it had the correct phylogeny that people had long predicted. And so this was really the motivation to say, let's go ahead and get one of these precious Neanderthal samples. And the sample that we got was from Vinja, Croatia, and it was a 38,000-year-old specimen that was found, a couple of, 20 feet down in this cave. And it was a bit of the femur, and it's basically a sequencing project. What you do is you mill, it's mineralized, you mill the material, you extract the DNA from it, and then you build libraries, and hoping that some of what you have there is really that ancient material. And basically it becomes a needle in a haystack problem, because you have a lot of sequence, and you're expecting that some of it is going to be what you're interested in, and then you use the power of blast to sift through all that to identify that which looks like hominid. And so, from the Neanderthal sequence, and we did this by using both capillary as well as 454 sequencing, 98% of the bacteria was plant, fungal, or environmental. And the best match we had sequenced some dirt a while ago, and that we had the most hits to that dirt sequence than any other, which makes sense because this bone was found in the dirt, and there were microbes in the dirt. But 1.3% of it matched hominid, okay? And a lot of work for only 1.3% of sequence, but the real issue is this really Neanderthal? And because once we can really prove that, then we can really go forward, because sequencing is, as I said, is getting cheaper, and 1.3% is a small amount, but you could sequence a lot for cheaply, or progressively so in the future. And so the way we proving that really was Neanderthal is crucial. There are a couple of ways we did this. One is showing there's certain DNA damage that occur. And the damage that occurs is cytosine deamination and fragmentation. And I'll show you how we use that in proving this really is Neanderthal. And another thing is looking at divergence time, looking at the sequence, seeing how it diverges from human sequence. And if we are able to show a big divergence time, then it's not two humans. Okay? And so the deamination, basically you lose this amine group with time. And the result of that is C's become T's, okay? And so C's are mutated to T's with this process. And so when we compared places where the human genome differed from the chimp genome, we looked at the ratio of C's from human converted to T's, and T's from humans converted to C's in the chimp genome. And what we see, this is a modern chimp, and we see a number, C to T and T to C are all the same. When we compared places where the human genome differed from our Neanderthal sequence, we had an excess of C to T, which is what one would predict if this is ancient. Another thing was, DNA is both sequence, it's also size. And we looked at the size of the DNA fragments. And the library inserts were 177 base pairs. The matches to hominid was 52 base pairs, small. We did the same thing, we did the cave bear, and likewise the cave bear had a small, with 67 base pairs, even though the library is a little bit larger. With the cave bear, we did have some contaminants that we were quite confident was modern human contaminants, and they were more than threefold bigger. And so the fact that in the Neanderthal specimen we don't have much hominid sequence out here is encouraging that this is really ancient and not a modern contaminant. Finally is divergence time, and this is when we align the human, the Neanderthal, and the chimp genome, most of it matches up, which is what you'd expect. Chimps are only 5-6 million years divergent. There was a small number of places where Neanderthal is different from human and chimp. I think lots of these are due to damage. We kind of ignored these. The ones we're really interested in places where human and Neanderthal were the same and chimp was different, as well as when chimp and Neanderthal are the same and human was different. And the fact that we got this sequence, it's hard to imagine contaminant leading to these kind of changes. And from this, we could calculate a divergence time, separating when the single human reference genome coalesced to the same genome that this single Neanderthal specimen is. And we get a time of 700,000 years ago, which is way longer than we'd expect to converge coalescence of any two human genomes. And so this is data strongly supporting that this really is ancient. The next question is, so the data suggests we have Neanderthal DNA. Is it a significant fraction of what we have? And we don't really have an answer to that. But we do have some evidence suggesting that probably most of what we have is really Neanderthal. And that comes from looking at the size of the Neanderthal fragments and then looking at the C to T damage as well as divergence time. And what we predict is the contaminants should be out in the larger fragments, the modern human contaminants, while the authentic Neanderthal should be the smaller fragments. And we broke the DNA into two size bins and looked at the divergence time as well as the damage amounts. And we see the big is no different from the small in that we see the same amount of damage and the same amount of divergence time in the two, so supporting that probably most of this really is ancient. All right, so what are we learning from the Neanderthal sequence? And one of the things is you could see where it's distributed. And what I'm showing you here are the human chromosomes. In blue are the human chromosomes. And the height is how tall they are. I mean, how big they are. And the red is the number of Neanderthal hits to each of these chromosomes. And what we're seeing, it's basically correlates with the size of the chromosome. So we're getting an even, we're skimming the genome. And you look at these bones, you can't tell if they're male or female. From the sequence, we have Y sequences, and definitely this Neanderthal was a boy. In other ones, we looked at the kind of categories. We have genes, we have non-coding sequences, we have conserved non-coding sequences. But it looks like what one would expect if one was just skimming the genome. And it appears to be no bias towards a particular chromosome or sequence classes. Okay, some of the questions, and this is one of the questions that paleoanthropologists always love arguing about, and that is the multi-regional or single origin of Homo sapiens. And the multi-regional model is that there was lots of mixing that went on. And basically, in Europe, this would be, humans and Neanderthals, a human precursor Neanderthal mixed until eventually there was a modern Homo sapien. Okay, and the single origin, which really is the classical, or not the classical, but the contemporary view of how evolution works, is that there was a common ancestor, and Neanderthals were one branch that came off that common ancestor. Homo sapiens was the other branch. Neanderthals died out, and Homo sapiens eventually evolved and multiplied and took over the planet. And so, so one of the things is estimating the split time for the human and Neanderthal ancestral population. This is work done by Jonathan Pritchard. And this takes into account, looks at derived SNPs versus ancestral SNPs, as well as makes population models. And so though we're saying that the Neanderthal, the human reference genome, this one Neanderthal sequences have a most recent common ancestor 700,000 years ago, the split time is when the two populations separated and stopped significantly mixing. And we come out with a split time of around 400,000 years. And this is long before anatomically modern humans have ever been noted. And so this goes very much against multi-regional hypothesis and goes through the single origin hypothesis. Another issue is admixture, and the public is really interested in this issue. And we don't have an answer. but we do have some data. And one is really looking for non-derived, non-ancestral derived SNPs. One would expect to see in the Neanderthals European non-ancestral variation. And we don't see any of that. And so that suggests that this probably didn't occur. But we have we have 65,000 base pairs of sequence, and it may be somewhere in there. I think when we get the entire genome, we will be able to nail this issue definitively, whether there was mixing that occurred. So several critical questions have been answered. Genomic DNA can recover from the Neanderthal specimens, and we have metrics for assessing authenticity and contamination. What can be learned from the Neanderthal sequence? Our data says that human and Neanderthal lineages diverged recently, but prior to the emergence of modern humans, saying that modern-day humans are not a blend that emerged from mixing that occurred recently. And we see no evidence for admixture. And the real fun stuff is what comes next, and that is how do we use the Neanderthal to gain insights into human-specific evolution, as well as learn about Neanderthal biology. And so right now we have the human, we have the chimp, we have the mouse genome. And there are certain things that humans can do that chimps can't do. And some of those, the genetic basis for some of those things are what are human-specific sequence changes. But this is 6 million years. Hopefully, in not too far in the future, we will get the Neanderthal genome. And that will give us a population time of 400,000 years. And Neanderthals, humans definitely do things that Neanderthals never did. And we seem to have had this intellectual explosion that occurred probably about 50,000 years ago. And, and I'm sure, likely there are going to be sequence changes that explain that. So the availability of the Neanderthal genome will be able to give us candidates, changes that contribute to uniquely human traits. And we have a list of about 500 sequences that we're interested in looking at. To see, people have hypothesized that some of these sequences, FOXP2 may be playing a role in language. There are certain variations in it that we think occurred recently, but it'll be interesting to look in the Neanderthal whether that occurred. And there are a variety of other things, brain development, a lot of biochemistry. There are lots of gene regulatory sequences that appeared to undergo accelerated evolution in Homo sapiens. And we'll see whether that occurred after or before Neanderthals and Homo sapiens split. Okay, so how do we do this? How do we get those specific sequences? One approach is to extract DNA and then directly shotgun sequence it. And this is an approach that Svante Pääbo, the Max Planck Institute, and 454 Corporation are pursuing. And this is a stamp, a 2.2 euro stamp that the Germans love the Neanderthals. They found it. And they really didn't do that much in the Human Genome Project, but they funded now a Neanderthal Genome Project. Another approach, the one that we're pursuing, is to build libraries, and hopefully libraries from several different Neanderthals. And with the libraries, one can immediately shotgun sequence it, but one also can go into those libraries and without sequencing the entire libraries, pull out specific sequences. And that's sort of the approach that we've been taking. And the way it works is we capture specific sequences from the ancient DNA library. So what you do is you take probes, and these are sequences you want to pull out. This is a mixture that we get from the ancient DNA library. Here are the ones that we really want. You use hybridization, streptavidin and biotin, coupling, pull them down, and go through several iterative processes to pull out specific sequences that you're interested in from the library. And the advantage is this, that you don't have to sequence several different Neanderthals. You can capture the same sequences from several different libraries from different individuals. And we've shown that this has worked with the cave bear. We have a pilot project showing that suggests at some level it is also working with the Neanderthal. And so this is where we're going in the future. And so the field is moving forward. One of the challenges is these precious samples, and they are precious because I don't know how many more we're going to get. You don't want any; they have to be destroyed for this kind of extraction. And so we've been trying to develop techniques to amplify the amount of DNA you get from the same sample, and it's working. And so we've also been able to show that we can repair some of the damage. And there are new sequencing platforms. And this 454 is one machine. There's this new machine that we have. 454 produces 30 million base pairs a run. Selects is another machine that produces 1 billion base pairs a run. So there are these new platforms that require less material and can sequence a lot more. Finally, the goal is to get multiple specimens. And you'd like to look at the same sequence in several different specimens so you don't have to worry about damage. And also you feel confident that really is a Neanderthal sequence change and not just a contaminant. And so right now we have... I think we have two different Neanderthals right now that are on the way to becoming DNA. So what's going to happen in the future? I think we're going to get 1,000 human genome sequence and not that far in the future. And they're going to have a lot of phenotypes associated with them. I think we'll probably get a bunch of different Neanderthal sequence in not far future. And so what are we going to be able to do with this? I think it's what the sequence will do for us. It'll tell us about human-specific traits that emerged in the last 400,000 years. I think there's a lot of interesting insights into Neanderthals that we're going to get from this, that we're never going to get from their bones and their stones. We're going to be able to things like, whether they like spicy food. We'll get clues about whether they had hair in their back. As we eventually learn to read the human genome, the human will be the reference phenotype for the Neanderthals. And so from what we know about humans and variation in humans that lead to specific phenotypes, we'll be able to go back to the Neanderthal genome and sort of be able to speculate that they had this kind of soft tissue biology, some stuff that we're never going to get from bones and stones. And finally, we need to keep the public interested in science. And there is an enormous interest in Neanderthals. And so I think this is a painless way to teach evolution. And it's hard to argue with theories of evolution when you begin to get a sense for these common ancestors. And so let me stop now with this motto, extinction is no excuse. And the work I talked about, the enhancer work, was largely, was a collaboration between me and Len Pinocchio and a couple of talented postdocs, Axel Wissel and Dava Tuv. And the Neanderthal work was really driven by a postdoc in my lab, Jim Noonan and Doug Smith, and it was a really lovely, wonderful collaboration with Svante Pabo, who got the specimens for us, and Jonathan Pritchard from the University of Chicago that helped in the analysis. And so I'll stop now.
Thank you for a great seminar. I'm sure I'll be happy to field a few questions.
All right, you.
So one of the obvious questions from the enhancer studies is that the 47% that you're capturing, those are all necessarily positive enhancers, things that are turning something on. Have you thought of any assays to detect the opposite of that, things that turn things off?
Yeah. No, we have, and it's hard. It's easier to do a positive than a negative, and so there are various. So we are developing negative assays, and we plan to try to go through. We can build constructs that look for, silencing activity in them, and so we plan on going through our positives and see if they're bifunctional. And there's polycomb. There people have identified polycomb binding sites, and so we do plan to pursue that.
I actually have a very similar question. Does the promoter that you use for that, I presume, provide some target information and some ascertainment bias into what you're going to identify? I mean, if you use a minimally active promoter, you're only going to get positive elements.
Right.
But presumably there's more subtle things as well. That overwhelming number of neurological promoters, does that have to do with the basal promoter that you use?
Yeah, no, that's a very good question. I don't think it's a promoter. I think people had a lot of experience with this promoter. We have a lot of negative controls where we see we've tested a whole bunch of things that had no conservation, and they never work as enhancers. And so, but the reason I think we see and so we're probably missing things because this promoter is not driving them, enabling them to be driven in certain places. But as far as, I forgot what was the first.
If you swapped out a different basal promoter.
Why there was neuronal? Yeah, I don't think we'll see much. I think, we have used, this is a heat shock promoter. We have used other promoters. And many of them we do, we do see similar, most of them we see similar patterns. Why we see mostly neuronal, and that is an ascertainment bias, because most of these ultra-conserved elements tend to be near genes involved in early development, and genes that are known to be expressed in central nervous system neuronal. So I think, and so that's why I think we're seeing so much neuronal. So I think we're really sifting through a certain category of sequences that we can pick up in our assay, because we can see those. Things that are involved in later development, it's somewhat hard to look in 11.5-day embryo. And so, but we really wanted a crank, and so we weighed it all, and we eventually came to this path. And so we're missing lots of things.
So you were able to stack like 4 enhancers, and they work additively. How many do you think that you can put together before you're too far away for it to matter anymore? Like
Too far away? What do you mean too far away from?
Well, in other words, how long do you think you can keep? Could you put 8? Could you put 16?
Yeah, I don't know. I don't know. I think we're going to stick at 4. Call it a win and walk away. But I think the way these things work is that I think these elements. Yeah, I think these elements have, in fact, though I showed you in space, these elements all have time activity. On the one with 4, the time activity was also recapitulated. And so I think the way these elements work is that I think that they are binding sites. And if there's not another transcript in front of it, if there's not another promoter machinery in front of it, it will cover the distance and turn the gene on at the site that information has predetermined. And so I don't know how many, but there are lots of examples where we have genes where there are lots and lots of enhancers in front of them, and so I don't see a reason why we shouldn't be able to recapitulate what occurs in nature. So I think 816 would also work.
Okay. Last question. So presumably about half of the sequence that you got from the Neanderthal was some kind of repeat.
So
Right.
do you notice any differences in the repetitive elements?
Yeah, we threw out all the repeat. If we had a sequence that hit more than one place in the genome, we'd throw it out because we couldn't really use that in coming up with human-specific or its match. We couldn't get a correct match, and so we didn't really pay attention to repetitive sequences.
Okay, well, I'm sure there are lots more questions. We'll have to draw it around so people leave. Please come up and tackle it one-on-one later.
Good to see you. I'll be at your next board meeting.